Good afternoon. Welcome everyone to our very first virtual ovarian cancer research tour at London Health Sciences Centre. We are delighted to have you all join us today for this very exclusive behind the scenes tour. Today, we are going to experience an insider's view of the labs, hear from a fantastic lineup of speakers who will be broadcasting live from their various locations, learn about the exciting ovarian cancer research happening right here in our own community, and most importantly, hear firsthand the inspiring impact of donor support. Thank you for your generosity, your passion, and your interest in partnering with us to help us improve patient outcomes. My name is Tracy Burwell, and I am a development officer at the London Health Sciences Foundation, together with my colleagues and the ovarian cancer research team joining us today. We are grateful to have you with us. I'd like to let you know a few things about today's tour before we get started. First of all, everyone is currently muted to protect the sound during the presentations. Secondly, this session is being recorded, and throughout the tour, we encourage your questions. Please feel free to write them in the Q&A chat button at the bottom of your screen, and we will do our very best to answer all of your questions at the end of the presentations, or you can expect follow-up with you offline. Thank you again for joining us and for your interest in this vital program. Now, I'd like to welcome you to my colleague, Erin, who will be introducing you to our guest speakers.
Hi, thank you so much, Tracy. So my name is Erin Pierce, and I'm a development officer at London Health Sciences Foundation. I'd like to echo Tracy and just thank everyone for being able to join us today. And we have a very exciting program lined up. So we'll get right into it and start with our first panelist, Dr. Gabrielle Dimatia, who is the interim director of the London Regional Cancer Program and co-lead scientist of the Mary and John Knight Translational Ovarian Cancer Research Unit. His research focuses on clear cell cancer of the ovary and specifically how the epigenome functions to direct cancer cell viability under different conditions relevant to this disease and all ovarian cancers. Dr. Dimatia is examining how these cancer cells maintain viability under low oxygen concentrations in different culture conditions, and if this form of ovarian cancer will be susceptible to drugs which inhibit the activity of the epigenome. Today, Dr. Dimatia joins us live from his office to share an overview and introduction to the Translational Ovarian Cancer Research Unit. Welcome, Dr. Dimatia.
Thank you, Erin. You pronounced my name very well. Congratulations. Good start. And hello to everyone out there in Zoom land. Welcome to today's presentation. I come to you today as the interim director of the Cancer Research Laboratory Program, and I'm going to give you a bit of a background of who we are and where we are. The Mary and John Knight Translational Ovarian Cancer Research Unit is embedded within the Cancer Research Laboratory Program. First of all, a very big thank you to all our supporters and fundraisers, and especially Jim Olson and the whole Run for Ovarian Cancer Executive Committee, who has put on this run for many, many years consecutively with great success. Thank you very much. Our gratitude is deep and steadfast. So I want to go on and tell you about the Cancer Research Laboratory Program. We're a group of 14 scientists. Most of us work in what we call wet bench laboratories, but we also have two labs that work in the area of bioinformatics. And that's bioinformatics with regard to image analysis, cancer imaging analysis. And that's all I'll say about that. But I'll tell you that we have foci of cancer research in a number of different areas: breast, head and neck, gastrointestinal, pancreatic, lung, and of course, ovarian cancer, all types of ovarian cancer. So we're located on the 4th floor of the London Regional Cancer Program at Commissioners in Wellington. We occupy about 28,000 square feet of research space. And that's made up, as I said, of 14 different labs. And we have on average between 120 to, let's say, over 150 personnel working within this research space. And within those individual labs, we're composed of high quality personnel, research technicians, research associates, and very importantly, graduate students at the MSc level, at the PhD level, and also MD PhD level, as well as undergraduate students at various years from Western, most of which are what we would call our fourth year honor students, all embedded in our laboratories. And I'm emphasizing this for you to make sure that you're aware that we're also a teaching and training facility. So research can't run without graduate students. They work very hard on their projects in order to obtain their degrees. And all of that work goes forward in publications from the various labs here, and we're very productive in that way. I want to also thank you again for your support indirectly of the Cancer Research Laboratory program. Your support of the Translational Ovarian Cancer Research Unit is critical. As you may know, the LHSC and any hospital in Ontario cannot directly support research with Ministry of Health funds. So all of the facilities that we have in the Victoria Research Labs and the Cancer Research Laboratory program on the 4th floor of the LRCP is all funded through the extensive work of the London Health Sciences Foundation and the Lawson Health Research Unit, which is our governing organizational body. You know, the funds that you have provided to support Trevor and his team have been instrumental in Trevor being very successful at obtaining outside funds and establishing really important scientific collaborations throughout Canada and the world. You may or may not know, maybe Trevor's going to talk about the fact that he was recently funded by the Canadian Institutes of Health Research. He got a research operating grant, which is a huge success, given that the funding rate is on average in the single digits to the low double digits at CIHR. He also has a very strong scientific collaboration relationship. He's one of the co-leads. He's a co-lead there of the Ovarian Cancer Research Program through the Ontario Institute for Cancer Research in Toronto, and has many different collaborations at Western and beyond. And your support has helped him to leverage local monies to get that outside financial support for his research. So thank you very much for that. And I think I'll sign off there.
All right, thank you so much, Dr. Dimatia. That was a lot of insight in terms of how the research lab works and the number of personnel who are working so hard day in and day out. So I'd like to introduce our next panelist is Dr. Trevor Shepherd, who is the co-lead scientist of the Mary and John Knight Translational Ovarian Cancer Research Unit and an associate professor in OBGYN, Oncology, and Anatomy and Cell Biology at Western University. Dr. Shepherd has received his PhD at McMaster University and has been actively conducting ovarian cancer research for the last 20 years. The major focus of Dr. Shepherd's research lab is to identify new ways to more effectively kill ovarian tumor cells while they are spreading. He's joining us today live from his office with an overview of what this looks like at the Mary and John Knight Translational Ovarian Cancer Research Unit. Hi, Dr. Shepherd.
Hi, good afternoon. Thank you very much, Erin, for that introduction. And thank you, Gabe, for giving an overview of our program as well. So what I'm going to do for this presentation, I'm actually going to share my screen, kind of give an overview of the focus of my research program. So just take a few seconds to get going. Just be patient. All right, excellent. So I'm guessing that everybody should see my opening screen here. And what I'll be describing to you today is the focus of our research program for my lab within the Translational Ovarian Cancer Research Unit. And that is to understand the mechanisms and the biology of how ovarian cancer spreads in patients. Because if we understand this biology, then we may find better ways to treat late-stage disease. So we think of ovarian cancer as actually having a unique mode of metastasis, or a unique way that it spreads in patients. And I say unique because most cancers will actually spread through the lymph nodes and the bloodstream. But in ovarian cancer, it most commonly spreads through the abdominal cavity. And that's seen where cells from the primary tumor that's growing on the ovary will actually break off and spread into the abdominal space. These will not only just be single cells, cancer cells, but they actually cluster together to form these 3D structures that we call spheroids. And this is something that our lab has been studying for about a decade now. And the biology of these spheroids is critical because it allows these ovarian cancer cells to actually survive while they're spreading and also evade cytotoxic chemotherapy. One of the other things that happens too within these spheroids is that they actually change their biology, and it enhances their ability to attach and start growing as new tumors on the surfaces of different organs in the abdominal cavity. It's also important to recognize that this is likely not just a one-way direction of spread, but that these new tumors that are forming on the surfaces of different organs in the abdominal cavity also have the capacity to, again, break off, form these spheroids, and continue to propagate late-stage disease. So if this is the way that the disease is spreading in a patient, how can we really test this using experiments in the lab? So to do this, over the last decade, we've developed a very special cell culture model where we can actually grow ovarian cancer cells in the lab in a way that mimics the way they may actually spread in a patient. So to do this, we use ovarian cancer cells, either new cells from patients right here at the London Regional Cancer Program from that malignant ascites, or that fluid that is removed at surgery or in the clinic, as well as other what we call cell lines, which are ovarian cancer cells from patients that may have been removed from a patient 10 or 20 years ago, and we can still use those cells in the research lab. Typically, these cells are grown in the lab by attaching them to tissue culture plastic in dishes with liquid growth media to allow these cells to grow and survive. But the way we grow these cells, ovarian cancer cells, to model metastasis is we actually use a special type of tissue culture plastic that has this what's called a hydrogel that covers the surface of the plate, and it doesn't allow ovarian cancer cells to attach. So it actually forces them to try to grow in suspension, kind of like how they would be behaving as they spread in a patient. And then we can model the last step where they're able to grow again after reattachment by just plating these spheroids, these three-dimensional structures, back into standard tissue culture plastic. Now, what we can do in this cell culture model is we can take these patient cells, these ovarian cancer cells, grow them in suspension to form these spheroids, and we can actually study their biology, isolate the genes and the proteins within these spheroids, and try to understand what are the changes that are occurring that might allow these ovarian cancer spheroids to survive and evade chemotherapy. Down below here are just some microscope images of what these cells look like in each of these different culture conditions, just to give you an example of what these ovarian cancer cells look like and what these spheroids actually look like. So over the last decade in studying ovarian cancer spheroids, what have we actually learned about the biology of ovarian cancer? So what I've done here is sort of summarize the general changes that occur in ovarian cancer spheroids and how this may impact the disease. So generally speaking, ovarian cancer cells, when they form these spheroids, they're actually under a lot of stress. And these stresses take the form of a number of different things. It's either the lack of energy, a lack of oxygen, lack of nutrients, and all of these together, these stresses, lead to what are considered starvation-like conditions. And ovarian cancer cells will then slow down their growth in order to try to survive, even though they lack these nutrients and oxygen and energy. So all of these factors taken together lead to sort of what we define ovarian cancer spheroids as a model of tumor cell dormancy. And what I mean by that is that ovarian cancer cells are actually in a way hibernating. So they're actually slowing down their growth and almost sleeping while they're spreading. And this is critical for two major reasons. One is we know that the chemotherapy that's used to treat ovarian cancer patients will actually attack and kill actively dividing cells. So if these ovarian cancer cells are actually undergoing this dormancy within these spheroids, that may be a mechanism that they're actually evading chemotherapy and they aren't being killed. The second is that this dormancy phenotype, or this hibernation state, allows ovarian cancer cells to actually maintain themselves for long periods of time and then emerge later, months to years later, potentially in a patient, to give rise to recurrent disease. And this recurrent disease is oftentimes resistant to chemotherapy. And we know that ovarian cancer spheroids are already somewhat resistant to chemotherapy. So this may actually be a mechanism that drives chemo resistance. So really this leads us to some major overarching questions that drive a lot of the research in my part of the research lab. And that is, how do dormant ovarian cancer cells handle stress? What are the changes that are occurring that allow them to survive in suspension and to survive this stress? And this is important because these stresses and these mechanisms by which ovarian cancer cells actually survive this stress, these could be considered Achilles heels of ovarian cancer cells within spheroids. These cells may be at that precipice, and we just need to figure out how can we push them over that and kill ovarian cancer cells more effectively during late stage disease. So I've broken up our sort of research projects into two major areas that I'll take you through over the next couple minutes. So the first is, again, this idea that as ovarian cancer cells spread within spheroids, or when they're treated with chemotherapy, they actually become stressed out. So we're trying to understand what are the genes and the proteins that are activated, that are turned on by this stress to actually help ovarian cancer cells survive. I'm just going to turn on this video so you can watch this as I continue to describe what's going on. So we're actually testing the different ways to block this stress survival response in ovarian cancer, and specifically in ovarian cancer spheroids. And we do this by trying to block the activity of these genes and these proteins that we've identified are critical within spheroids. And that's shown over here in this video that was actually captured using our IncuSite Zoom. And this is where we've actually used a technology called CRISPR, which is a molecular technique that goes in and deletes specific genes or cuts out specific genes out of the genome and stops them from functioning. So in this case, we've CRISPR deleted a single gene that we've identified as critical in ovarian cancer spheroids. And you can see that it reduces the ability of these spheroids to grow and survive. And they're actually breaking apart and breaking down in this spheroid culture model. And as Gabe already mentioned, this is actually a group of projects that we're working on right now that is now being funded just recently by a new grant from the Canadian Institutes of Health Research. One of the other major projects that we have going on in the lab that's related to this stress response, but it's very specifically looking at a process called autophagy. Autophagy means self-eating. And this is a natural way that cells in our body can actually survive starvation-like conditions, where they will actually break down some of the molecules within a cell to recycle them and create new nutrients and more energy to survive these starvation-like conditions. And we've identified that spheroids actually activate or turn on autophagy to survive when nutrients are running low within spheroids. And that's just shown over here in this static image of a spheroid, where we're actually using a special, what's called a fluorescence reporter. It actually shines as a specific color of light that gives us an idea that a certain pathway or process is being activated. And here, what's shown in red are all the cells within a spheroid that are actually activating autophagy within this spheroid. So what we're doing now is, again, looking at genes and proteins and pathways that are being activated to promote autophagy. And can we block this alone or in combination with chemotherapy to more effectively kill ovarian cancer spheroids? And again, as Gabe mentioned, this is a project that we've been working on for a few years now, and it's been funded by the Ontario Institute for Cancer Research. So just for my last slide, I want to highlight the members of my research team that are doing a lot of this work that I just described very briefly in the last slide. I want to point out our research manager. She actually runs the day-to-day operations of our translational ovarian cancer research unit. She's co-supervised by both Gabe and myself, and she helps to train all of the trainees for both of us, as well as I said, helps to support a lot of the research activities of the lab. Also, a research technician, Olga, who has many years of experience doing ovarian cancer research, first in Ottawa, and then we recruited her to work in our lab. Tharsha is a new research assistant that helps in procuring new patient specimens from the operating room as well as from the clinics. And then all of our trainees. So postdoctoral fellow, Dr. Adrian Buensuceso, PhD candidates, Matt and Jacob, and MSc candidates, master's thesis candidates, Emily, Dolly, and Jack. And these are the individuals who are actually doing all of the research that I've just described very briefly on the previous slides. I'd like to point out, as Gabe had mentioned as well, that a lot of the work that's being done in our lab has been funded either in the past or currently by funds from the Run for Ovarian Cancer, as well as numerous other donors to our program. We're very thankful for that. Just as an example, I'd like to point out that both Jacob and Matt, their projects are actually currently funded by LRCP Catalyst grants, which use funds from the Run for Ovarian Cancer. And with that, I will end, but I will just actually highlight that 3 of these trainees you will see over the next half hour or so with their own presentations on their projects, as well as some of the technologies that we're using in the lab. So with that, thank you everyone for joining us today, and I'll turn it back over to Erin to introduce our next speaker.
Thank you so much, Dr. Shepherd. It's really fascinating to learn how ovarian cancer differs from other cancers and what the lab is doing to try to understand that and combat it. So just a reminder, if anyone has any questions for any of our panelists today, you can submit them to the Q&A at the bottom window of your screen in Zoom, and we'll be answering them at the end of all of the presentations. So our next panelist is Emily Thomas. Emily has received her undergraduate degree from Western University with an honors double major in microbiology and medical cell biology. She has completed her first year of her master's degree in anatomy and cell biology at Western, and has been working at the Translational Ovarian Cancer Research Unit under Dr. Shepherd for 10 months. Emily's thesis project focuses on using a new mini tumor model called organoids to help researchers better understand how ovarian cancer cells adapt to form tumors and survive during metastasis. And she is joining us today from the Graduate Student Office. Welcome, Emily.
Hi, everyone. I'll just get my presentation going here. Yeah. All right, so thank you, Erin, for the introduction. And today I'm just going to be giving everyone a little bit of information on my master's thesis project, which is on three-dimensional mini tumors as models to study ovarian cancer. So just to kind of reiterate what Dr. Shepherd was talking about earlier, so ovarian cancer, it's a very complex disease, as we know. There are many different types, But a majority of patients are diagnosed with the most aggressive type, which is high-grade serous ovarian cancer. And this is the type that I'm going to be focusing on for my project. And also, like you said, it is a very unique disease because cells are able to break off from tumors and form these small clusters called spheroids within the abdomen as the cancer metastasizes. And with this, it also kind of causes this buildup of fluid within the abdomen. And when it contains these malignant clusters or spheroids, it helps them easily spread. And so overall, this makes it really difficult to treat patients. So a lot of our research is done to better understand this entire process so that we can help improve treatment options for patients. So for my project, I am using these two 3D cell culture model systems, and I'm comparing how the cells behave within each model to better understand ovarian cancer. So the first model here is the organoid model, and this is a new model being used in cancer research to try to study tumors. And how it works is that it creates the right environment in this kind of solid matrix dome as the cells are cultured in this little matrix here. And it allows the cells to develop into these mini tumor structures. And then we can observe the cells within these structures to better understand how tumors form, how tumors grow, and how they are organized overall. Now, the second model is the spheroid model system. And like Dr. Shepherd said, we have used this model for many years to try to understand ovarian cancer metastasis. And just again, how this model works is we culture the cells in this fluid, and it allows them to be suspended in this liquid, and it allows them to form these clusters or spheroids. And this is to learn how the cells are adapting under stress during metastasis. And the whole reason that I am using these two models is it's because we believe that cells are able to switch. They have this kind of switching behavior from tumors to spheroids, and then potentially back again as they form secondary tumors in the abdomen. So by using these two models, I can directly compare the cell behavior and the cell biology within a tumor and within a spheroid to gain more knowledge on ovarian cancer and potentially find targets within this process to help prevent spread. So my project aims to answer kind of that big pressing question on ovarian cancer on switching behavior. And to do this, I am growing cells within these two models and observing them as they grow and develop, and then collecting them afterwards to do many other experiments and using a bunch of different technologies within our lab. And I just wanted to highlight that an important part of this project is to also make sure I generate translational results. And this is part of our whole research unit goal to make sure that our results accurately represent patient characteristics and account for any differences seen between patients. So I'm actually using patient-derived samples, which can be developed cell lines or actual cells collected from patient tumor samples at the London Regional Cancer Program here at Victoria Hospital. And so I have a few aims that I want to accomplish for my project as well. And first is I want to determine the appearance or morphology of these cells, because how they look definitely ties into how they behave within each model. And second, I want to determine what these signaling events or which signaling events are critical for tumor growth or metastasis or both processes. And to do this, I'm identifying the specific signals or activated proteins that cause certain cell behaviors or adaptations. And third, I want to dig a little bit deeper and see if I inhibit any of these important signals or proteins, if that will kill the ovarian cancer cells or at least inhibit their growth to help prevent spread. Now, why is this project important? So the results from my project will have a substantial impact on ovarian cancer research because firstly, it's going to help us further understand how ovarian cancer behaves and hopefully identify the changes that occur during the whole progression of the cancer from tumor growth to metastasis. And it will also help us to establish this new model for our research. And with this, how to accurately use each model, the organoid model or the spheroid model, for any future experiments on ovarian cancer if we want to look at specific signals or specific proteins. And lastly, once we understand these models, we could potentially use either of them or more the organoid model as a platform for therapeutic testing, which kind of ties into this idea of precision medicine, which is a growing kind of field in cancer research. And to do this, we could take a portion of an ovarian cancer tumor, grow the cells as organoids, which are seen in this image here, and then test the cells with a variety of different treatments to see which treatment is going to work best for that specific patient. And this is really important because it could eliminate the potential for drug resistance following chemotherapy or using non-specific therapies that don't necessarily work for all patients. So that's everything. I just want to thank you for listening to my presentation. I would like to thank everyone in the lab and all of our collaborators, as well as all of the women who are donating their tissue samples for our research and this unit, and as well to everyone who has graciously donated to the London Run for Ovarian Cancer event.
Thank you so much, Emily. That was a very interesting presentation. Excellent thesis project. Our next panelist is Dr. Adrienne Buen-Sucesso, a postdoctoral associate in the Mary and John Knight Translational Ovarian Cancer Research Unit. Originally from Montreal, he came to London for graduate school and has been a member of the research team for almost 6 years. His goal for his work is to find new ways of disrupting ovarian cancer cells' ability to adapt to stress in order to impede cancer spread. Dr. Buen-Sucesso is joining us from the lab and will be guiding us through how he uses three-dimensional cell culture and automated microscopy to study ovarian cancer.
So can you guys hear me just fine?
Yep.
Okay, awesome. Thank you. So like Erin said, I'm a postdoctoral associate here at the Mary and John Knight Translational Ovarian Cancer Research Unit. I guess if I had to describe it in a nutshell, the work that I do in the lab focuses on understanding the mechanisms by which ovarian cancer cells adapt to stresses during metastasis, and finding ways that we can disrupt those mechanisms for therapeutic purposes, for therapeutic benefit. Now, as you might guess, as a member of the research team, I pretty much spend most of my day doing experiments and analyzing data. And so part of what I'm doing today is setting up to grow ovarian cancer cells under conditions that mimic the environment in many ways inside of a patient with metastatic ovarian cancer. So to start with, setting up these experiments is almost all done inside of a unit that's called a biological safety cabinet, which is this large unit right beside me. So really the purpose of this piece of equipment is to keep unwanted microbes out of our experiments, bacteria, fungal spores, and so on. Now I have all of the materials that I need inside of the safety cabinet behind this glass shield. So really safety first, it's important. I need to make sure that I'm wearing my lab coat and that I'm wearing my gloves. And any object that I bring into this safety cabinet, including my hands, has to be sprayed with ethanol in order to kill any microbes that might contaminate my experiment. So for the experiment that I'm setting up today, first off, I need liquid growth media, which provides the cancer cells with nutrients. So that comes in bottles like this. And I'm going to give it a quick spray with ethanol. Make sure I cover all of the surfaces. to kill any microbes that might be residing on the bottle because it's coming in from the outside. And I also need to bring in tools that I use to accurately and precisely move liquids as well as cancer cells between different kinds of vessels. So one of those tools is called a micropipetter. So I need to spray that down as well. All right, so my colleague Bart is going to just kind of try to maneuver the laptop and webcam so that you can more closely see what I'm trying to do here. So working inside of this biological safety cabinet, I have a tube that contains medium along with ovarian cancer cells that I will be growing over time. And the cell culture vessel that I'm using looks like this. It's really made of plastic that's specially treated in order to prevent cells from attaching to the surface. It really keeps cells in suspension. And the reason that we want to do that is because it simulates the environment in a metastatic ovarian cancer patients where cancer cells have shed from the tumor, they've detached from the tumor and are sort of suspended in ascites or other kinds of fluids. This micropipetter, takes these disposable tips that actually come into contact with the medium and the cancer cells. And what I'm going to do here is just transfer some of the cells that I have sort of pre-allocated into this tube into my cell culture vessel. So if you can maybe like angle that a bit up. Okay, perfect. Thanks, Bart. So I'm very carefully allocating my cancer cells into this vessel. All right. So for the purposes of this experiment, I don't need to completely fill up this vessel. And so really the purpose of the experiment that I'm setting up today is I want to, you had heard Dr. Shepard mention the concept of ovarian cancer spheroids. So we use a number of different ovarian cancer cell lines in the lab. And so for this particular cell line, what I want to do is monitor spheroid growth over time. So monitor a spheroid size over a long period of time, potentially 2 weeks or longer. So in order to do that, we use an instrument called an IncuSight Zoom, which is essentially an automated microscope. So this is a unit that will automatically focus on the cancer cell clusters or spheroids in my experiment and capture images over a period of at least 2 weeks, and actually also perform some of the analysis in an automated fashion as well. So if we could just move over to the IncuSight over here. So the IncuSight Zoom lives inside of an incubator, which we use to maintain the cells that we're growing at a temperature of 37 degrees Celsius, 5% carbon dioxide, and an environment with saturated humidity. So I open up the incubator, and we have the IncuSight Zoom unit, the automated microscope in here. So I can just open it up and place my plate inside. Make sure it's sitting nice and securely. So you can see that there are some other plates in here as well that other people are using for their experiment. So one of the cool things about this piece of equipment is that we can have 6 different vessels in here being monitored at the same time. So potentially 6 different experiments going on simultaneously. And this unit can actually monitor our experiments, capture images, and many kinds of metrics that are important for the experiments that we do over long periods of time, but also frees up our time to do other things. So typically, I might have to take pictures of these cells using a microscope, doing that manually, taking my time. But this thing is always doing that even overnight and over the weekend. So it's great that it's always collecting data for us. So close it up, close up the incubator, and that plate is ready to go. So thanks a lot, Bart. Okay, great. So, of course, because I just set up this experiment, I don't have the data yet to show you, but I can actually kind of walk you through the kind of results that we would obtain from the IncuSite Zoom in another experiment or a previously performed experiment. So I'm going to try to do that right here. Share my screen. Okay, great. So I hope you guys can all see that. Okay, awesome. So really, the whole idea behind what I'm doing today, like Aaron mentioned, using 3D cell culture and automated microscopy to study ovarian cancer. So the overall workflow here is essentially to take our ovarian cancer cells, place them into appropriate cell culture vessels, like I had done inside the biological safety cabinet, and then transferring these vessels to the IncuSite Zoom automated microscope. And so you might be wondering, what kind of data, what kind of results do you get out of the IncuSite? So this is really just showing over on the right a sampling of images of ovarian cancer spheroids captured over a period. This experiment actually went out over 2 weeks, but for the purposes of being able to fit all of this on one PowerPoint slide, I've chosen 3 of those time points. So day 1, day 7, and day 10. So you can see that the spheroids are actually growing and getting larger over time, but we need an objective way to make that statement. So we need an actual way to measure the size of these spheroids. So one of the nice things about the IncuSite is it can perform this automated analysis so in order to determine spheroid size over time. And so you can see that the IncuSite has accurately masked the spheroid in orange over in this bottom set of panels. And so when we've accumulated all of this data over a long-term experiment, so this is the graph showing on the y-axis spheroid size over time, which is on the x-axis. This is actually many different time points. I think we had something like almost maybe 50 time points here. And you can actually see that over time, we're able to track the size of the spheroid and get an accurate measure or estimate of its growth potential. So, this is just one of the types of experiments that I might set up on a day-to-day basis. Yeah, I hope you enjoyed this sampling or this slice of a typical day in my life here in the Marion John Knight Translational Ovarian Cancer Research Unit.
Thank you so much, Dr. Boed-Sucesso.
Of course.
That was very exciting, and it's great to see your research in action. So we do have one more panelist for today, and then we're going to be moving on to our Q&A session. So if anyone has any questions, you can submit them at the bottom of your screen just by using the Q&A button, and we will answer them at the end. So our final panelist is Matthew Borelli. Matthew is a second-year PhD candidate, a recipient of the Karen Pooley Scholarship in Ovarian Cancer Research, and a member of Dr. Shepard's team in the Marion John Knight Translational Ovarian Cancer Research Unit. He has completed an undergraduate and a master's degree at Western through the Department of Physiology and Pharmacology, and has recently been awarded a National Doctoral Scholarship from the Natural Science and Engineering Research Council of Canada, which will fund the remainder of his PhD research. Matthew is investigating metabolism in ovarian cancer, more specifically, which metabolic pathways are altered in spheroids and the molecular mechanisms that produce these responses. He's presenting today live from the office on energy stress during the spread of ovarian cancer. Welcome, Matthew.
Thank you, Erin. I'm just going to get my screen shared here. And let's hop over into PowerPoint. Okay, so thank you again for the introduction, and hello everybody. First off, I want to say thank you for attending this tour today and for joining me for this presentation. Today I'll be talking about some of the work I've been doing to investigate responses to energy stress during the spread of ovarian cancer. I'd also like to add that I would be more than happy to discuss my work with you and answer any questions or curiosities you may have. So please don't hesitate to send your question through Zoom to be answered after the presentation. I will say I am going to be getting into basically this presentation is going to be really examining one of the pathways that I've been researching. So there are going to be a couple acronyms thrown around, but I'll try to keep it fairly understandable. But even still, please feel free to ask any questions and I'd be more than happy to answer them. So to start off, I'd like to touch on ovarian cancer spheroids, as we've been going over today, as an experimental model, since these structures are very important in ovarian cancer metastasis, and therefore they are a critical component of our lab's research. Unlike most other cancers that metastasize by invading the bloodstream, you've heard that ovarian cancer metastasizes by forming these spheroid structures, which are free clusters of cells that are able to detach from a primary tumor growing on the ovary and travel through the abdomen to form metastases or new sites of disease at different sites. To study spheroid biology in vitro, we've also gone over this, we simply seed cells into ultra-low attachment culture plastic, and we allow the spheroids to form spontaneously over the course of hours to days. Here are a couple examples of spheroids from cell lines I work with. These are different cell lines, of course, just to show sort of a sampling of the different types of spheroids that we can end up seeing, and really giving you an idea of the heterogeneity or the level of difference that we can see across different cancers. So we see here, this is an example of a very large, dense spheroid that we get from culturing HA8 cells. That's one of our cell lines. And the IOVCA 147 cell line was actually derived from a patient who donated her ascites to our research program. And we see that this line forms much looser, less dense sort of clumps of cells that don't really form this bulky structure like the HA8s do. So for cells and spheroids, floating freely without a solid base to attach to is highly stressful, as Dr. Shepard mentioned. And under normal circumstances, these conditions activate a process called Anoikus. Anoikus is a type of programmed cell death that is activated specifically when cells are no longer able to attach to a solid substrate. In a real case of ovarian cancer, the substrate that the cells would be detaching from in this analogy would be the tumor, the primary tumor that's growing on the ovary. In our lab, cells are normally grown on plastic dishes, so this is where they detach from in our in vitro or experimental models of ovarian cancer metastasis. Since spheroids are able to stay alive for long periods of time, we understand that the cells that they're comprised of must be adapting in some way to overcome this stress that would normally lead them to undergo anoikus. And what we do is we study the adaptations to this stress. I'll mention a couple of important ones that our lab has worked on in previous years. The first is that cells in spheroids shift to a state called quiescence. You can think of quiescence a lot like hibernation, as Dr. Shepard mentioned. So energy-intensive activities like cell division and maybe protein production are temporarily limited in order to conserve energy to survive as long as possible. So effectively, the cells are able to go to sleep in a reversible way such that they're able to wake up again with viability, alive, and able to reinitiate the cancer at a different site. Second, in spheroids, we see activation of a protein that's called AMPK. And AMPK is a critical player in the process of slowing down metabolism in spheroids. And in addition to shutting down metabolism, or in addition to shutting down growth signals and slowing down metabolism, AMPK induces a recycling process called autophagy that Dr. Shepard also mentioned earlier. Autophagy, which literally translates to self-eating, allows cells to recycle a variety of their own contents in order to sustain their energy stores and survive for longer. Autophagy in spheroids is another area of active research in our lab. I'm going to be talking a little bit more about AMPK going forward, though. Before I joined Dr. Shepard's lab, when our lab first started to investigate the importance of this protein in spheroids, they tried performing a gene knockout of the protein that normally activates it, which is called LKB1. And the knockout was successful, and it had these really profound implications for limiting the survivability of these cancer cells as spheroids. But what was interesting was that while the knockout was successful, AMPK was still being activated. So what that meant was that AMPK activation in ovarian cancer spheroids doesn't happen through the normal mechanism. Closer to the time that I joined the lab, we started looking at another protein that can activate AMPK, which is called CAM kinase beta, which I'm going to refer to as CAM going forward. Using a drug inhibitor of CAM and by making CAM knockout cells, we've been able to definitively show that this is the protein activating AMPK in ovarian cancer spheroids. The next question that we want to answer is why? Because we know that calcium signaling is the predominant signal that activates CAM kinase beta and therefore would be activating AMPK, but we don't understand yet what the actual role of calcium in these spheroids is. So that's something that we want to look into further moving forward. So moving forward, I'll be investigating how the loss of CAM and therefore the loss of AMPK activation affects ovarian cancer spheroids. So taking a look at some early data that I've generated so far, looking at cells that lack CAM, which are referred to as CAM kinase 2 KO, the results are pretty interesting, and we see that there's some differences between different cell lines again. For reference, these images of spheroids, as with the previous ones, were captured using the IncuSight Zoom system you learned about from Adrian in the last presentation. And this was over a period of 1 week for these images, and the further images are going to be up to 2 weeks as well. These spheroids are still actually being imaged now. They're still in the machine that Adrian opened up every 3 hours. So it's really incredible what we can do with that machine. So taking a look at the images themselves first, when we look at a day 7 spheroid for an OVCAR8 cell line in comparison to the CAM knockout cell line, we don't see that there's all that much of a difference, except maybe the CAM knockout looks a little bit rougher around the edges. However, if we look just 3 days later at 10-day spheroids, the CAM knockout has almost completely fallen apart while the normal cells remain intact. And it's completely possible that this might be due to the loss of AMPK activation that we see in these spheroids. On the other hand, it's clear already from day 7 that looking at day 7 HEA8 spheroids, CAM knockout results in a smaller spheroid that is able to grow much slower. Fast forward a week though, and the normal spheroid has begun to disintegrate while the knockout stays intact. One thing that I want to investigate going forward is trying to determine why these cell lines between the OVCAR8 and the HEA8, for example, respond differently to this same gene knockout. My second focus is on an organelle called the mitochondria, which some of you may have heard referred to as the powerhouse of the cell. And they're so named because the mitochondria, or the mitochondrial network, is a major source of energy that cells need to survive. I've included an immunofluorescence image here, which is a microscopy technique where I've actually zoomed in on our cells at approximately 1,200 times magnification. And this type of, or this immunofluorescence microscopy allows us to visualize different components of cells that have been stained for specific markers. Each blue blob that you can see here corresponds to one cell, so it's one blob per cell, and this blob that's being stained is the nucleus, which is where the cell stores its DNA. This erratic web-like staining that you see in the red color is the mitochondrial network in each of these cells, and you can see that each cell has between tens and hundreds of mitochondria. In the top left corner, I've also included a black and white inset showing the same image in its processed form. So I've written a macro in a program that we use called Fiji, which allows me to determine the size of each individual mitochondria present in any image that I want to. So then I can compare across different images, across different cell lines, and between adherent cells and spheroids. So without getting into too many details with respect to the mitochondrial work, so far it's been aimed towards identifying differences in the organization and the function of mitochondria between spheroids and adherent cells. And with respect to that, much of the data that I've gathered suggests that in spheroids, mitochondria are broken up into smaller pieces inside the cells through a process called mitochondrial fission, smaller in comparison to the adherent cells. My hypothesis right now is that this process might be occurring in order to make the mitochondria more bite-sized, as we refer to autophagy, in the sense that maybe smaller mitochondria are able to be consumed by autophagy to produce more energy for the cells. And with that, I'd like to thank Gary Pooley for very generously supporting the research I was able to touch on today through the Karen Pooley Scholarship in Ovarian Cancer Research, as well as the funding from the Marion John Knight Translational Ovarian Cancer Unit. And finally, the incredible support we receive through all of you and through the London Run for Ovarian Cancer. Thank you all so much for supporting our research and for joining us today for this tour. For now, I'm going to hand it off to Erin, who I believe will be kicking off our question and answer period. Thank you for your attention.
Thank you so much, Matt. That was very fascinating. Yeah, so as Matt said, we're going to move into the Q&A portion of the day. I would like to invite Dr. Shepherd back. If you have any questions for any of the panelists, you can submit them now using the Q&A function at the bottom of your screen. We will try to get to all the questions, but if we don't, we will reach out to follow up. So we'll start off with our first question. So one of the participants is wondering if there's testing available to determine if you have a genetic predisposition to ovarian cancer. So for example, if your mother had ovarian cancer, are there genetic tests that would determine if a daughter is a carrier? Thinking this would perhaps mean more careful tracking to attempt to catch ovarian cancer before stage 4. Dr. Shepherd?
Yeah, that's a great question, obviously. So in terms of genetic testing and seeking the advice of genetic counselors, yes, the LRCP has that. And within our gynae oncology disease site team, they have Karen Panabaker and Peter Ainsworth. They lead the genetic counseling team here. So right now, from what I have seen in my interactions, it's typically women who are diagnosed with very specific types of ovarian cancer can be referred to genetic counseling to get the testing done. So this will either be high-grade serous ovarian cancer because there's a high prevalence of having mutations in BRCA1 or BRCA2 or other genes that can predispose to developing high-grade serous ovarian cancer. There's also another type of ovarian cancer that is due to what's called Lynch syndrome. These are mutations in other genes as well, and that can also be identified through genetic testing, as well as through pathology. And those women can be more predisposed to ovarian cancer, uterine cancer, as well as colon cancer. So that's another sort of genetic predisposition to developing ovarian cancer as well. In terms of just a general seeking, genetic testing, because you've had a family member in the past who's had ovarian cancer, that's where I think you would want to consult with genetic counseling to really understand whether or not that would be supported through funding to actually get that genetic testing, or whether or not there would have to be additional factors. So that's my answer from a non-clinician, but from what I've heard, that's typically how it would be.
Okay, perfect. Thank
Do
you.
you mind if I chime in?
Yeah, for sure.
So here at the LHSC, we have a molecular diagnostics lab, which is led by Dr. Beckham Sidikovic on the 10th floor of the new building we have here. And it is one of the premier molecular diagnostic labs in Canada. And in fact, Beckham was trained here in the cancer research laboratories with Dr. David Rodenheiser when he was here. And he has set up a number of custom genetic tests to look specifically for mutations in the genes that Trevor just pointed out. And so those are available, but of course you would have to talk to the genetic counselor, and the genetic counselor would also take a family history, and that family history plays a big part in determining whether further genetic testing is required. I hope that helps to answer the question.
Oh, you're muted, Erin. There you go.
All right, so our second question is, how do PARP inhibitors work, and has the work in our lab contributed to developing them?
Yeah, so maybe I can go first with sort of explaining a little bit on how PARP inhibitors work. So the premise here is that, again, that most prevalent type of ovarian cancer, high-grade serous ovarian cancer, is a very what we call genomically unstable tumor. So a lot of the DNA is broken up into different fragments, and there's a lot of changes in the DNA. So what that does is it actually, in these high-grade serous ovarian cancer cells, there's a lot of defects in DNA repair, mainly due to those mutations, as I already said, in BRCA1 and BRCA2. So what research has shown over probably about 2 decades now or more, that because of those defects or mutations in BRCA1 and BRCA2, high-grade serous ovarian cancer cells are actually, it's like one of these Achilles heels. They already have these defects in DNA repair. So the cell really requires other DNA repair proteins to try to survive and try to maintain the integrity of the DNA. And one of these enzymes is PARP. So what has been developed are new drugs that can actually target PARP and inactivate PARP. And these would be effective in tumors that have mutations in BRCA1 and BRCA2, because now it's like a 1-2 punch. They've already got this mutation in BRCA1 and BRCA2. They really rely on PARP to keep that DNA intact. And if you treat a patient with that PARP inhibitor, it will more effectively kill high-grade serous ovarian cancer cells. So that's the reason why right now it's actually approved for use in women with mutations in BRCA1 and BRCA2 with high-grade serous ovarian cancer, as well as breast cancer. But where clinical trials are actually going is that it may actually be effective for even a greater number of women with high-grade serous ovarian cancer, because there may be other defects related to BRCA1 and BRCA2. And that's where PARP inhibitors are being used there as well. My research, and Gabe can pipe up as well. Like, our research did not contribute to PARP inhibitor development. That was started, back when I was actually doing my postdoc work. Labs were already jumping on PARP inhibitors, and it wasn't just for ovarian cancer. Like I said, it was breast cancer and that as well. So it took some time to develop, but now it's approved for use in ovarian cancer. But our work didn't lead to that.
Okay, thank you. So our next question is, how do ovarian cancer cells become resistant to chemotherapy, and why does this happen more with each chemo treatment?
Yeah, that's a great question. So there are hundreds of labs trying to understand, like, how do ovarian cancer cells become resistant to chemotherapy? So there are many different ways that cancer cells can do this in other cancer types, and some of these are actually used in ovarian cancer as well. So some of these mechanisms could be that the cancer cells themselves are able to pump those drugs out of the cells. So the drug gets in, but then there are molecules or proteins that can actually pump that drug back out of the cell. Another mechanism could be that the drug gets into the cell, but the cell has figured out ways to survive that toxin, has figured out ways to upregulate other genes and proteins to actually live even within that toxic environment of chemotherapy. Another way as well is that tumors can actually change the blood supply and change their metabolism. So now those chemotherapeutics can't really work the way they're supposed to. And probably the best answer for the idea of why is it that with every time that chemotherapy is used, it becomes less and less effective, you can think of it as a process of natural selection. These tumor cells, there are billions and billions of tumor cells within a growing tumor. And every time chemotherapy is used, it's killing 99.9% of those tumor cells. But now you're selecting for the cells that have figured out ways to evade that chemotherapy. So when the disease comes back, it's less and less likely to be killed by that chemotherapy.
I'll just add to what Trevor, my take on that is what Trevor said, chemotherapy certainly can kill the vast majority of cancer cells. But to go back a little bit earlier to what Trevor talked about in terms of PARP inhibitors, one of the major hallmarks of high-grade serous ovarian cancer and other cancers like pancreatic cancer, head and neck cancer, triple-negative breast cancer, is that the DNA or the genome of those cancer cells is highly disrupted. It is constantly undergoing chromosome breaks and reattachments. And that is a mechanism that indirectly allows cancer cells to change their identity, if you will. So a tumor is not made up of a single type or single identity of cancer cells. It's actually made up of probably hundreds, if not thousands, of different identities. The genome of those cancer cells, even some that are right next to one another, is different. And that difference in their gene expression and the proteins that they produce, as Trevor alluded to, allows some of those to grow faster and evade the killing mechanism, if you will, of chemotherapeutics. And the hard part for cancer researchers and clinicians who treat cancer patients is that this process never stops. It just keeps going and keeps going and keeps going. And that's why the work that Trevor does on dormancy and understanding how we could kill those dormant cells is very important, because those cells harbor their own genetic mutations that allow them to become dormant to some extent and then take off again when the chemotherapeutic pressure is released, when chemotherapy treatment ends.
Okay, thank you very much.
That's great.
All right, so another question we have is, what other technology besides the IncuSite do you use in the labs?
Yeah, that's great. So obviously we had a short virtual tour of the lab. We would have loved to take a camera and go around the whole lab and show different pieces of equipment and what their capabilities are and why we use them and how we use them. We do that when we do the live tours, and I know everybody really enjoys that component. So there are many different pieces of equipment. So Adrian showed you the IncuSite, the incubator, and the cabinet there that we are able to work with cells. But in other rooms of our research unit, we have what are called PCR machines. So PCR, my father-in-law likes to call it like the photocopier of DNA. But basically, it's a way where you can amplify DNA, pieces of DNA, and you can double it every few minutes. So you can go from one piece of DNA to billions of pieces of DNA just within like an hour. And these units are actually done in a way kind of like with the IncuSite, where you just put your reaction into the machine and you walk away. And not only is it performing that experiment, but it's also quantifying that DNA over that 1 hour period. So it's a way where you can use minuscule amounts of starting material and you can analyze hundreds of different things within just an hour. And this is a technology that was developed when I was a graduate student. I think Gabe was probably a postdoc graduate student around that time or even before. And
I was a graduate student too.
Yeah, okay. And I was very early graduate student. And basically, this technology started where, it was a very laborious process to be able to do PCR. It took hours, keep adding enzymes and more components to the reaction. And it has developed to the point now where everybody can do PCR very quickly and inexpensively for almost any sort of DNA or RNA-based molecule. So that's one piece of equipment. We have a lot of different microscopes. Matt showed you some data using confocal microscopy. That's something that we don't necessarily have just for our own lab. We've got a couple different confocal microscopes that are used, as Gabe mentioned, for the whole cancer research laboratory program that people have access to get those really high resolution microscopy images. And then of course, you can do a lot of quantification of that as well. We do have microscopes that are really dedicated just to our translational ovarian cancer research unit that we were able to purchase from donor funds a number of years ago, a number of different fluorescence microscopes and light microscopes that we can capture a lot of really important images of our cells and spheroids. And I could go on and on. We have many different pieces of equipment. Some of which are, yeah, Gabe, go ahead.
No, I just wanted to interject when Trevor talked about those microscopes to send a thank you out to Jim Olson, because and a note, because of course his late wife Mary was responsible for obtaining the funds to buy those microscopes that we use each and every day. And I'd also like to say that it's not just our lab that uses these microscopes. There are many labs within the Cancer Research Laboratory program that come to us and ask to use it for their work as well. So, we're spreading that great wealth amongst all the groups here, and it certainly facilitates our work as well. And I think Trevor would like to probably say something about the IVUS bioluminescence system up in the vivarium as well.
Yeah, absolutely. Yeah, can't believe I forgot about that. So in the last couple of years, we also were able to purchase, and again, this is a significant piece of equipment that we're able to share with other research labs in the Cancer Research Laboratory program here at Victoria Hospital. It's housed in the mouse vivarium on the 7th floor of the VRL, or the Victoria Research Labs. And this is an instrument. So we didn't actually talk about it today, but we talked mainly about our cell culture work. But one of the next steps of doing ovarian cancer research and a lot of cancer biology is to take it to an animal model. So this is important to try to understand, the next order of complexity of how cancer grows and spreads. And we do that with ovarian cancer. We'll actually take ovarian cancer cells and we'll put them into immunocompromised mice. And we want to be able to see how do these tumor cells spread. It's important to be able to visualize this over time. Now with ovarian cancer, just like in human patients, it's actually very difficult to visualize and measure this in a mouse, because it's difficult to see where are those tumor cells spreading, where are they growing, until the mouse is getting very sick with advanced disease. But with the IVIS Lumina bioluminescence instrument that we have in the animal facility, this gives us the ability to actually visualize in mice tumor growth and spread non-invasively over time. So you can actually track how are those tumor cells spreading. And importantly, we've already started doing experiments where we can test some of these new drugs that can target a process called autophagy and knock out some of these different genes, put those cells into mice and see, can we slow down the growth of ovarian cancer? Can we block metastasis from occurring? So this is, this was an instrument that we really wanted to have years ago, and we were able to get the funding to be able to purchase that equipment and from donor funding. And like Gabe said as well, we're able to offer that to other research labs, and many different labs are starting to use that, including ourselves.
Perfect, thank you. Our next question is, at the high level, what does the 5-year research roadmap look like, recognizing that it will be results driven?
Gabe, do you want to start out with that one?
Well, I mean, I think, is that from Gary? I think Gary's talking about your CIHR grant, Trevor, because that's a 5-year CIHR grant.
Okay, right. Yeah, so if that's what it entails, so the CIHR grant is specifically focused on those stress pathways that Adrian is working on and that Matt talked about as well. So it's the idea of LKB1 and CAM kinase beta, and how they actually are controlling the survival and the adaptation of ovarian cancer cells within spheroids, and then to take that to animal models. So yes, that's a 5-year program, and there are 3 different research aims for that. One is to look at how does this stress responses affect inflammation within ovarian cancer cells, because that can be one of the adaptations that occurs when ovarian cancer cells are spreading, that they actually are sort of in an inflamed state. And inflamed cells will change all the different molecules that they're secreting to try to promote their survival. And we think we have data to support the idea that these stress pathways are actually promoting inflammation. So that's a lot of the work that Adrian is working on right now. The other aim is really focused on a lot of stuff that Matt talked about, and it's this metabolic reprogramming of ovarian cancer cells. How do ovarian cancer cells change their energy state during this time of stress? And can we target that by both hitting different genes and proteins, and potentially even with different therapeutics? And then the last aim is actually then taking it to these organoids that Emily talked about, as well as to mouse models of metastatic ovarian cancer, to see that those inflammation phenotype and the energy changes, energy metabolism, are those things that are actually going on during natural progression in an animal model. And that's to support both the operating costs and to do those experiments, as well as to support some of the personnel to do that.
Okay, great. Can you speak a little bit about the impact that the instruments and technology and things like that you have in the lab, the impact those have had on cancer?
I guess like on the
I think they're interested in the AMPK and things like that.
On the clinical impact.
Yeah.
Yeah, so I think obviously translating this to potentially clinical trials and eventually to clinical care, that can be years down the road. Where I think the research that we're doing right now that is most closely aligned to that translational component and a clinical component is what Emily talked about. The idea of working with cells that are taken right from ovarian cancer patients that are being treated here at the LRCP. So it's either from that ascites fluid that's collected, as well as for those organoid models to actually take small pieces of tumor and actually grow them as organoids in the research lab. So that's a project that has those direct sort of translational implications. That would be my answer to that.
Well, you also just recently did an MRT study in the xenografts. I mean, that's pretty close to and the work that you've done with OICR in defining the pharmacokinetics of that drug and so on.
Yeah, so what Gabe is referring to is this new drug compound, relatively new drug compound that can block autophagy, a very specific protein that controls autophagy. And right now we're doing that instrument, we're using that IVUS instrument and treating ovarian mice with ovarian tumors with this MRT compound and seeing whether or not it can actually affect tumor growth as well as spread in mice. And this is part of that OICR funded program to actually test these new compounds that might actually have clinical relevance. Sort of the preliminary data that we have right now is that it's definitely effective in vitro in our spheroid culture model system. We're still processing the data from the mouse studies. My take on it is that even if it's promising that this MRT compound on its own would probably have to be optimized. And then that would be, again, further work with the drug discovery program at OICR to actually make this drug sort of clinical grade and to optimize it for use in patients. And then that would lead into phase 1 and phase 2 clinical trials, that sort of idea.
I think it's important also to recognize in answering that question that remember that cancer therapy these days is not a single drug for any single disease. It's usually a combination of drugs. And the work that Trevor's doing is one where perhaps the initial hypothesis or question was, I think he alluded this too in his discussions here, can this approach weaken high-grade serous ovarian cancer cells to the point where those more harsher chemotherapeutic compounds, carboplatin and Taxol, can be more effective at wiping out the burden of disease in women carrying ovarian cancer and allow for a much longer progression-free survival and also for multiple rounds of therapy in order to extend progression-free survival.
Okay, great, thanks. So we have one final question here. In terms of precision medicine based on a specific patient's ovarian cancer cell type, how close are we to putting this technology into practice?
That's a great question. So really, we have examples of that happening already. So obviously the PARP inhibitor, that is an example of precision medicine. It's the idea, like right now, it's approved for use in women with BRCA1 and BRCA2 mutations, and it's shown to be quite effective at prolonging survival. So, so that's an example of precision medicine, taking, basically the genetic fingerprint of that tumor and being able to identify what would be the right drug combination to more effectively treat that patient and their tumor. Are there other examples of that in ovarian cancer? There are definitely clinical trials that are specifically focused on that. So it gets back to some of the comments that were made by the trainees in their sort of introductions, that we understand that ovarian cancer is actually not just one disease. There are multiple different types of ovarian cancer. And we understand that it's actually driven by very specific mutations for each one of these different types of ovarian cancer. And that's where the field is really pushing now, is that we need to move away potentially from carboplatin and paclitaxel, or at least having it where there will be new therapeutics that will work better in combination with carboplatin and paclitaxel based on, is it low-grade serous cancer? Is it high-grade serous cancer? Is it clear cell mucinous or carcinosarcoma? And it'll be based on those mutation signatures. And there is, I believe it's province-wide, there is what's called Octane. And it's the idea of actually fingerprinting patients' tumors, not just for ovarian, but for many different tumor types, fingerprinting genetically those tumors, and then being able to actually put those patients on very specific therapeutics that will be more effective based on that fingerprint.
One of the interesting things, this question, we think about answering this question from a gene level, from a genomic level. But in fact, I would say 30 to 40 years ago, precision medicine was already underway, specifically in breast and prostate cancer, with the development of anti-androgens and anti-estrogens like tamoxifen. So if a woman came in with a particular type of breast cancer that was estrogen receptor positive, she would also be given tamoxifen to inhibit estrogen action, which drives the growth of that tumor. The same with prostate cancer. If they were androgen positive, men would be given anti-androgens. So that's a form of precision medicine. And it is actually pervasive now in oncology. A new donation in the pancreatic cancer world here locally, the Baker Center for Pancreatic Cancer has instituted that sort of testing of pancreatic cancer tumors to look to see if there are specific targeted drugs out there that may help these patients, for which there is not much in the way of treatment these days. So certainly that's happening, as Trevor eloquently put, in ovarian cancer, but it's across the board. It's a very, very big push.
Okay, great. Thank you very much, Dr. Shepard and Dr. Dimatia, for answering those great questions. I'd like to introduce back my colleague Tracy, and she's going to do some closing remarks.
Thank you, Erin, and to everyone who posed those awesome questions. And thank you to our incredible ovarian cancer research team for your time and presentations today. Wow, we are so very fortunate to have your expertise right here in London, Ontario. To all of you, our donors and partners, the RUN committee executive, event participants, thank you for your inspiration to invest in this life-saving research. Everything we've seen and heard today wouldn't be possible without you, our donors. We are so grateful that you've joined us today to see firsthand the impact that you're making. Donor support is vital to everything we do. And that concludes today's tour, wishing all of you and your families a happy and healthy summer ahead. Thank you again.